purpose of this video is to walk students through the online Fly Lab for the Science Courseware site. These examples will show how to identify linked genes and calculate recombination frequencies. Linked genes are genes that are located on the same chromosome. Most AP questions will ask students to analyze data and identify patterns of inheritance, which is why it is so important to do the Fly Lab. In order to complete the crosses, you will need to navigate to the sciencecourseware.org website. Select the Fly Lab JS, which stands for JavaScript option, and on the web page you will see displayed several options. Students can select between male and female, and then select from several traits. Once the trait is selected, the different phenotypes will be displayed above the fly. The female will have the wild form of the traits, which is long wings and red eyes, and the male will have the mutant form of the trait, which is vestigial wing and sepia eye. The offspring of this cross are called the F1 generation, and they will exhibit the dominant form of the allele, and the genotype will be heterozygous for all the F1 offspring because both parents were homozygous for their respective phenotypes. The test for a linked gene is to perform a test cross. This is done by selecting an F1 female hybrid with an all-mutant male fly from the parent generation. If genes are on different chromosomes, then independent assortment and crossing over will occur during the formation of gametes, and there will be 4 resulting phenotypes and equal proportions of each phenotype. For the chi-square test of this data, be sure you have clicked the box Ignore sex of flies because we already know that this is an autosomal trait, and click the box Include a test of the hypothesis in order to enter your expected ratios of offspring. A chi-square analysis is used to calculate the variation from the 1:1 to 1:1 ratio. Notice that the chi-square value is much less than our critical value of 7. So this means that we will fail to reject our hypothesis, and these 2 traits of wing size and eye color are unlinked and located on different chromosomes. In this cross, the data will first give you information about the mutant traits of sepia eye and spineless bristles. In the remaining crosses for this lab, you will follow the same format, producing an F1 hybrid and then performing a test cross using the F1 hybrid and the male mutant fly from the first generation. When the genes are linked and on the same chromosome, there will be way more parental phenotypes than recombinants. Parental phenotypes have the same combination of traits as the original P generation flies, and the recombinants will have new combinations of the traits. Since the original flies from the parent generation were wild for both traits and mutant for both traits, we will be using the flies in the second and third row of our data table to calculate our recombinant frequency. The gene linkage is confirmed by performing a chi-square test of the data. As in all the prior crosses, for the linked gene portion of the Fly Lab, the expected ratio and also null hypothesis is a 1:1 to 1:1 ratio. In this cross, the chi-square is much larger than our critical value of 7, and this means that we reject the null and conclude that the genes are linked and located on the same chromosome. Now that we know that genes are linked and on the same chromosome, we can use this data to calculate the recombinant frequency. It is important to remember that the reason why there are such low numbers of recombinants is because there is only crossing over as a source of variation from the parental phenotypes. We can calculate the recombinant frequency by dividing the number of recombinants by the total number of flies. In the claim, make a clear statement about the inheritance pattern. If you fail to reject the null, the traits are unlinked. If you reject the null, the traits are linked. For the evidence, there also needs to be a statement about the chi-square value. This is normally followed by a description of the phenotypes and the ratios of offspring for each phenotype. If the genes are linked, a recombinant frequency can also be given as a part of the evidence. The reasoning is where you relate which concepts of genetics relate to the results observed. Be sure to stop and pause to only include those concepts that actually relate to the cross performed. Examples of concepts that can be related to this portion of the lab include differences between linked and unlinked genes, the sources of variation for phenotypes, or lack thereof, details about the relationship of the recombinants to the map distance, and how these values are used in the making of the gene map. An example of a statement that could be included is, in the F2 generation, there were way more parental phenotypes than expected and way less recombinant phenotypes than expected. This is because the only source of variation is crossing over when two traits are on the same chromosome. Be sure to go back and review the important vocabulary listed. This is Ms. Wilson speaking and wishing you a golden day. I hope this video helped you retrieve some helpful information.